Okay, so I'm going to start with this experiment, which is how to stain a leaf for starch. It's fairly straightforward, so you just have to memorize the steps, and you also want to understand what each step is doing. All right, so you might remember from the food tests that the reagent that we use to test for starch is iodine, and it turns blue-black in the presence of iodine. It's originally kind of a brown rust color, right? And so the idea is that the first thing you do is you take a leaf, all right, and you are going to boil it in water and just for maybe 30 seconds to a minute. And the reason you do that is that it removes the wax, the waxy cuticle, right, from the upper part of the leaf. We talked about the waxy cuticle, so it removes the wax. That way the iodine can actually penetrate into the leaf. Right? Then after you boil it in water, you're going to boil it in ethanol. And remember that ethanol has a low boiling point, so you need to be very careful with this, okay? So usually this is done in a water bath or after the Bunsen burner has been turned off after you've boiled the water, okay? Um, and the boiling the ethanol, so you boil in ethanol for a few minutes until the leaf becomes kind of yellow or colorless. And so what you're doing is that removes the chlorophyll. And the reason you need to remove the chlorophyll is because that allows you to actually see the color change. That green that is that leaves have from the chlorophyll can mask any changes in colors in the iodine. All right. So the idea is that in ethanol, you boil it in a water bath that removes chlorophyll. Okay, then you take it out and you rinse in cold water and you spread it out. And that kind of softens the leaf, but they hardly ever ask about why you're doing it. So you rinse it in cold water, and then you add a few drops of iodine. Okay, and this is to actually stain for starch. And so the idea is that if starch is present, then the iodine will turn blue or blue-black. All right, so those are your 4 steps for how to stain a leaf for starch, okay? And this only works if the leaf has been left out in the light for several hours, because then it has done enough photosynthesis to create starch, right? Remember that starch is how plants store their carbohydrates. So the glucose that's produced from photosynthesis is stored as starch. And so if you want to destarch a leaf, okay, if you want to make sure that at the if you're doing an experiment where this would be the readout, right, where you set up an experiment and then at the end you want to see whether starch is present in the leaf, the first thing you would want to do is actually put the plants to destarch for before you begin the experiment. So I'm going to add that at the end. Okay, so to destarch a plant or a leaf, all you have to do is make sure that it can't photosynthesize. And what does it need for photosynthesis? Light. So you put it in the dark for, I don't know, 12 to 24 hours beforehand. And so the idea is that because it can't do photosynthesis, it's not producing any glucose. So it will break down the starch that is there in the leaf to use for energy and respiration and things like that. So the idea is if you, oops, I already had that. Haha. Anyway, so to destarch a plant, you put it in the dark for maybe 12 to 24 hours. And that's going to make sure that when you start your experiment and your test, you can test very different things. In this, you could look at the effect of different wavelengths of light. You could look at the effect of temperature on how whether starch is produced, keep them at different temperature. You could do all sorts of things like that. Then the idea is that you would want to start out and make sure that the plant has no starch at the beginning. And to do that, that's how you would do it, destarch it.